you to the students the questionnaire. It's okay? All the students. Okay. Good morning everyone, and welcome to the stem cell month. With the first month of the year, we start to organize each month characterized by a topic. For January, we have stem cell. So this is the program for all months. Today we have the first mini-symposium about stem cell biology and application. So it is an introduction on stem cell. Next week, in January 19, we will have a new mini-symposium about organoids culture. And then on January 26, we will have a webinar. Okay, we can start today. The mini-symposium, stem cell biology and application, will be presented today by Valentina Mastroiaco, Serena Riccitelli, Luciana, Gneo, and Ponzetti. And it will be organized in this way. Valentina will present you in introductory part about stem cell, the pluripotent stem cell and tissue stem cell. Then we will see the application of stem cell in research, in clinical, and for new drug discovery. So, start.
Good morning, everyone. Today I will present you an introduction about stem cell. So, first of all, what are stem cells? We say that stem cells are the foundation cell of our body. In many tissues, they serve as a sort of internal system repair, dividing essentially without limit to replenish other cells. Also, they are not yet specialized. Why stem cells are so special? First of all, we distinguish stem cells for other cell types for two important characteristics. The first of all, they are undifferentiated cells, are capable of renewing themselves through cell division, sometimes after long periods of inactivity. In fact, when a stem cell divides, it can replicate itself and give rise to another stem cells, but also they differentiate into many cell types. In fact, under certain physiological experimental conditions, they can be induced to become tissue or organ-specific cells with special function. Also, such as muscle cells, blood cells, and brain cells. In some organs, for example, gut and bone marrow, they divide regularly to replenish other cells, and also in other organs, such as the brain, the stem cell only divide under special conditions. So, we know four kinds of stem cells derived from human and animal, which have been studied by the researcher, and these cells are embryonic stem cells, adult or somatic or tissue-specific stem cells, induced pluripotent stem cells, and cancer stem cells. First of all, we describe embryonic stem cells that are described as pluripotent, meaning that they give rise, they can generate all different types of cells present in our body. They can be obtained from blastocysts, that is a very early stage of development, and then these cells can be grown and expanded indefinitely in an undifferentiated or unspecialized state. This ability helps researchers to study early human developmental processes that are otherwise inaccessible, and also help the researcher to study disease and to establish therapeutic strategies designed to replace and to restore damaged tissues. Likewise, they can be coaxed to differentiate to a variety of major specialized cell types, and this is a challenging tool for regenerative medicine. The other kind of stem cell are adult stem cells that are already somewhat specialized. In fact, they can produce some or all of the major tissue and the major cell type found in a tissue. For this ability, they are described as multipotent. As we can see in the first picture, in adult brain, the adult stem cells are able to give rise to neurons and to glial cell type, astrocyte and oligodendrocyte. Also, we can say that this kind of cells have been found in several organs that need to continuously replenish themselves, such as skin, blood, and gut. And also, they have been found in other less regenerative organs, for example, the brain. In fact, the primary role of the adult stem cells are to maintain and to repair the tissue in which they are found. These kind of cells are thought to reside in a specific area in each tissue called stem cell niche, and these cells are a very small population, often buried deep within a given tissue, making them difficult to identify, isolate, and grow in a laboratory setting. The main difference between embryonic and adult stem cells are these, and first of all, they have different ability in number and type of differentiate cell types they can become. In fact, as I said before, the embryonic stem cells have the potential to give rise to all the cells of our body because they are pluripotent. The adult one are thought to be limited to differentiating into specific cell type of their tissue of origin. The second difference consists on the growth in culture. In fact, embryonic stem cells are relatively easy to culture in spite of the adult one. In fact, they are rare in measured tissue, so isolating these cells is challenging, and methods to expand their number in laboratory setting have not yet been worked out. The last difference is on likelihood of being rejected after transplantation. In fact, adult stem cells and tissue derived from them are believed less likely to be rejected, to initiate rejection after transplantation. In fact, patient own cells can be expanded in culture, coaxed into assuming a specific cell type and then reintroduced into the patient. In this way, these cells are less likely to be rejected by the immune system, and also the patient could avoid the continuous administration by the immune suppressive drug, which may cause deleterious side effect. Another kind of stem cells are iPSCs, induced pluripotent stem cells, that are adult cells that are engineered or reprogrammed to become pluripotent. They have the same characteristic of embryonic stem cells, and in 2006, Yamanaka and colleague showed that the introduction of 4 specific genes in adult cells can be convert the cells into a pluripotent stem cells. And these 4 genes, as you can see in the box, are OCT4, SOCT2, CMYC, and KLF4. And this type of cells could be a powerful tool for studying the cause of disease for testing drugs. The last kind of stem cells is cancer stem cells that have been defined by the American Association of Cancer Research as a cell within a tumor that possesses the capacity to self-renew and to cause the heterogeneous lineage of cancer cells that comprise the tumor. In fact, they are a subpopulation of cancer cells that present a greater renewal potential compared to the other tumor cells, and also they have the ability to reform all the characteristics of the primary tumor upon transplantation into immunodeficient mice. Also, they have been postulated to responsible to tumor growth, to tumor relapse after therapy and metastasis, but they are only validated in breast cancer and in acute myeloid leukemia. So in conclusion, we need to identify an abundant source of cells, because identifying, isolating and growing the right kind of stem cell are painstaking and difficult processes. This kind of cells are so important because we can use them in a lots of applications, for example, basic research, clinical treatment, and drug discovery. Thanks for your attention.
Thank you very much, Valentina. For the discussion, we prefer to have a general discussion at the end of all the presentation. So now I will present to you Luciana.
Good morning, everyone. I'll talk about the role of the stem cell in the role of stem cell in human biology research. Today we still don't have proper treatment for many diseases like cancer or neurodegenerative disease, and the stem cell would be the solution for the treatment of these incurable diseases. Research on stem cell is advancing, and they can be used to basic research, to clarification of complex events that occur during human development, and understanding molecular basis of cancer or other disease. In biotechnology, stem cell can provide specific cell type to test new drugs, and in cell-based therapy we can use stem cell in regenerative therapy, in gene therapy and therapeutic cloning, and we can use stem cell to understand their role in cancer. Stem cells are undifferentiated and are capable of dividing and renewing themselves for long period. They are unspecialized and unconvenient until it receives a signal to develop into a specialized cell. And a primary goal of research on stem cell is to learn how undifferentiated stem cell turn into differentiated stem cell that form specific tissue and organs. And today I focalize attention on the embryonic stem cell. Embryonic stem cells are capable of becoming all of the various cell types of the human body, and for this reason it's a pluripotent cell, making them a promising source of cell for treating many diseases. Embryonic stem cells are derivated from the inner cell mass of the developing blastocyst. Embryonic stem cells present more advantages, are flexible, in fact have the potential to make any cell, lasting because can provide an endless supply of cells, and for their availability. In fact, we could use embryons from the in vitro fertilization clinics. But how to derive an embryonic stem cell line? Embryonic stem cells are derived from the cell in the inner cell mass of the developing blastocyst. And to grow stem cell, we can remove the inner cell mass and culture them in a dish in the lab. In this condition, stem cell growth and divide several times, and we can divide the population in two other dishes. And we can obtain two different culture. Stem cell line in the stem cell line when stem cell divide themselves but non-differentiate. And in the case in which stem cell differentiate and change in the cell condition, we can stimulate the cell to differentiate into a variety of cell type, such as neural, skeletal, or skin. In fact, one of the biggest hurdles in an embryonic stem cell-based therapy is causing stem cell to become a single cell type. And when we have embryonic stem cell in culture, we can grow them in a nutrient-rich solution, and the differentiation are induced in the presence of exogenous factors such as growth factor, interleukin, or cytokine. And in this way, a specialized stem cell gives rise to specialized cell in response to external and internal chemical stimulus. Another method that we can use to obtain a differentiated stem cell is a co-culture. In co-culture, we can grow in a dish embryonic stem cell and a monolayer of other cell type. In this kind, embryonic stem cell co-cultured on a monolayer of the bone marrow stromal cell line, the OP9DL1, that express an appropriate notch ligand, exhibit in a specific culture condition. In fact, in the medium, there is the presence of the interleukin 7 and the F83 ligand. In this condition, embryonic stem cell exhibit the ability to differentiate in most hematopoietic lineage, and in this case, we have the production of T cell progenitors. But the embryonic stem cell presents some limits. In fact, produce teratoms, a tumor with tissue or organ component, when transplanted into animals. Present high antigenicity. In fact, patient's immune system will recognize transplant cell as foreign and attack them. And when scientists isolate human embryonic stem cell in the lab, they destroy an embryo. So the ethical and the legal implication of this have made some reluctant to support research involving human embryonic stem cell. In recent years, some researchers have focused their effort on creating stem cells that don't require the destruction of embryos. And for this reason, we can obtain embryonic stem cell, and in particular, we can create patient-specific embryonic stem cells using therapeutic cloning. In therapeutic cloning, we can isolate the cell from the patient, take an egg cell and remove the nucleus from an egg cell and transfers the nucleus containing DNA from the patient cell to the egg. And in this way, egg cell reprograms the patient's DNA, begin dividing, and we can obtain a blastocyst and take and isolate the inner cell mast cell. In this way, in theory, the embryonic stem cell can become any type of cell in the body, and since these cells are made from the patient's own DNA, there is not a danger of rejection by the immune system. An important role is also the induced pluripotent stem cell. In fact, using this cell we can resolve the ethical and legal problems for the embryonic stem cell. Induced pluripotent stem cells are created artificially in the lab by reprogramming a patient Owen cell and can be made from readily available cell including fat. And also in this case we there is no danger that the immune system will reject them. In the end, a good application, it represents only the odor stem cell. In fact, odor stem cell exists naturally in the body and they are important for growth, healing and replacing stem. In fact, today stem cells from the blood and bone marrow are routinely used to attract treatment for blood-resistant disease. But odor stem cells are present in low abundance and are difficult to isolate and grow in culture. So, in conclusion, we can say that stem cell research has the potential to have a significant impact on human health. There is some controversy around the development, usage, and destruction of human embryos, and scientists may be able to ease this conscience by using a new method that can turn adult stem cell into pluripotent stem cell, which can change into any cell type. So despite this advancement, there is still a lot more to be done before scientists can create successful treatment through stem cell therapy. Thank you for your
Thank you, Luciana. And now, Marco. Will present a presentation about the drug discovery.
So, good morning everyone again. So today I'm going to talk about these human induced pluripotent stem cells and how they can be used to screen and discover new drugs. So first, what is a drug discovery? This is a process through which new candidate medications are proposed and tested. And these drugs can be both natural or synthetic. They can be classical, biotechnological, or phytotherapeutic. There are two main kinds of drug discovery processes that exist, and the first one is the stochastic process, and the second one is the hypothesis-driven. So the stochastic is basically what has usually happened many times in history. It's the serendipitous discovery of many drugs, such as penicillin. So you know that Pasteur found that his culture was contaminated and bacteria were not growing well. And then he said, okay, there should be a factor that is preventing the growth of the bacteria. And so he said, let's study this. And he discovered penicillin and saved billions of lives. The hypothesis-driven is different because you start from an hypothesis. So you have a target. I want to block this receptor. I want to use, I know the active site of the receptor, and I want to develop a drug that is specific for that application. So this is the difference. So here's another example of a stochastic discovery of a phytotherapeutic, which is a natural drug, which is also how I like to think that the pharmacology was born. So there was a caveman, he was sick, and he said, I'm so sick, I'll try to bite this tree. So after that, the caveman felt much better, because that was a willow tree, it was a salix, so he discovered aspirin. But despite that, many developments in the drug discovery since the cavemans, actually we still start from more than 10,000 different compounds just to get through this pipeline that leads to just one approved drug. So we start from 10,000 compounds and we end up with one drug. And this is very time and money consuming, because you see this process take more than 13 years. So, for example, this is a more in-depth example of a drug discovery process. There is a first phase, which is the research and development, in which you found your target of interest, you screen for different compounds, and then you identify a lead compound, which is a compound from which you start to optimize the formulation of the drug. And then you start the preclinical studies, which are made up of in vitro and in vivo studies. And there is many standards for these preclinical studies, and I have to say that these are not perfect. So many drugs arrive in clinical trials, which, as everybody knows, have different phases. They arrive here and then they stop at most of the phase 2 trials. That's what happens in most drugs. But if they manage to pass this phase, they then go through the review and approval process by the FDA in this case. But in Europe, we have a different organization that approves drugs. And this can also be very strict. So despite all these new tools we have bioinformatics, we have 3D crystallographies of targets, which we can use to perform molecular docking studies. We can repurpose drugs. We can use new drugs for old targets, old drugs for new targets. The lead compounds are very easily identified now because you can just analyze the active site via bioinformatics. And active compounds are very well predicted, but despite all that, the vast majority of drugs don't make it further than phase 2 clinical trials because of normally a lack of efficiency or organ toxicity. And why is that? Because we lack appropriate preclinical models. These preclinical models are usually either immortalized cell lines, which many of us have used in the lab. There is different cell lines that are used for different applications, but you should know that these cells are immortal. They are basically almost cancer cells in many issues. So you cannot say that these are normal cells. How can you say that these are a good model for a normal cell? They normally are a better model than anything, but they are not a perfect model. Then we can use rodents for in vivo studies. Of course, a mouse is very different from a human, but still, they're better than nothing. And then we can use larger mammalians for toxicity studies, which would be a better model than rodents, but they are far more expensive. So normally we start from rodents and then the larger mammalian only for specific studies. So even combining all of that, we cannot achieve more than 70 to 80% predictive ability. So another useful tool that we could use for drug discovery would be using the human primary cells. These are a powerful tool to improve the predictiveness, but they are often hard or impossible to obtain because think of obtaining neurons from a person. You should pierce into his brain. It's not that easy. They are often hard or impossible to grow because, again, thinking about neurons, they do not grow well in culture. They are parental cells. And when they are obtainable, they are usually few. So this is when the induced pluripotent stem cells come in the game. And they are coming in the game in the latest years because there are better pluripotency induction protocols that are being developed. There are better differentiation protocols. They are produced from a minimally invasive skin biopsy. They can be obtained by the patients themselves, so there is no risk of rejection. They can be grown extensively before differentiating them in perennial tissue, even in neurons. And they're particularly useful when trying to treat diseases affecting permanent or stable tissues, such as hepatocytes or the heart. So these are very useful in drug discovery also for complex diseases that involve more than one gene. I'm talking about genetic diseases. So you can take the fibroblast from the patient directly, induce them to the pluripotency, and then use them to study the disease. And they are also useful for sporadic diseases or with different somatic mutations. So, for example, for disease modeling, the cells have been used for spinal muscular atrophy, where the patient-derived iPSCs have been differentiated into motor neurons, And the research show that they degenerate much faster than the control iPSCs. Then there is a cardiac disease that is the type 2 long QT syndrome, in which these cardiomyocytes that are derived from iPSC show a prolonged action potential, and we